So you're live now.
All right, a very good morning, everyone. Welcome to all the students who are attending the MOOC courses on proteomics and proteogenomics. In fact, let me welcome all the participants, learners, everybody who is part of this course. We have really large number of students and participants from all over India. In fact, some part of the world also who participate in these courses. And we realize that given the significance of these omics technologies, and especially proteomics and proteogenomics, some of the tool and techniques and concept which we are describing in this course is going to be helpful for all of you, whether you work on the human or any other model organism in any sphere of the life sciences. These technologies can be very much applicable. Today we are doing this live session of MOOC course for both the groups of proteomics and proteogenomics. I'm Sanjeeva Srivastava, your instructor, which you have been seeing me in the video over the weeks. Today you have opportunity to talk to me and ask me any questions in between. But to give you the flavor of how these technologies work, we are going to conduct this session from our laboratory today, so that you can also get an opportunity to see how these experiments can be done using these technologies. So to make the session more interesting and focused, today we have chosen this topic, mass spectrometry based clinical proteomics applications. And before I actually go further about today's session and talk about some of these concepts, and we show you these technologies, let me first introduce some of the TAs who are working very hard to run this course. They have been taking your queries on the portal. And let me first introduce for the proteogenomics course, Mrs. Shalini Agarwal. Shalini has submitted her PhD thesis now from IT Bombay. And she has been working on malaria, which is one of the still major problem, vivax malaria, using proteomics and some of the proteogenomics tools. Let me show you this introduction clip of Shalini.
Good morning all. My name is Shalini Agarwal and I'm a PhD scholar at IIT Bombay under the guidance of Professor Sanjeeva Srivastava. Work of my PhD revolves around the diagnosis and prognosis of Plasmodium vivax in the infected host, that is human. So here I'm going to give you a brief background about what I do. So I collect the whole blood samples and divide the whole blood sample into two parts. One is the host plasma and the other is host RBCs. So these parasites invade RBCs. So to understand how the proteome of the parasite is working, we need to extract the parasites and then we study the parasite proteome. And after analysis, we try to get few of the parasite biomarker proteins which can be used as a diagnostic candidate. So those candidates which were highly potential biomarker candidates, we had taken those and cloned and expressed it in the bacterial system here, as you can see in this particular part. But in the case of Plasmodium vivax, the thing is that the parasitemia is not directly proportional to the severity of the disease. Hence, we need to understand the host protein as well. So we had taken plasma sample and we tried to understand the significantly altered proteins as compared to the healthy participants. And then we performed artificial neural networks and models on the data set what we had to predict or to understand the proteins which can be predictive biomarkers. Combining these two diagnostic and prognostic candidates, we tried to come up with an assay which can have these two components together to get an idea that a patient is Plasmodium vivax malaria positive and whether it's going towards severity or not. So the idea is to create a diagnostic kit in future, which can be taken forward to clinics to get an accurate diagnosis of Plasmodium vivax infected patients and whether they are moving towards severity or not. This will help us in getting a better sample cohort, which can further lead to better understanding of the disease and how the pathogenesis is happening. So that is all for my work in brief. So I'll be TA of the course Introduction to Proteogenomics. And throughout this particular course, I'll be there to help you all If you have any question, you can post it on the forum as you all have been doing till now. And I'll be there to answer your queries. So hope to hear from you soon and happy to interact with you. Thank you.
All right, thank you, Shalini. And Shalini has been the TA for Proteogenomics course for last couple of years. She's quite experienced. As you can see, she is doing the research in this direction. So of course, you have more opportunity to interact with her. Let me now introduce to the next TA, which is Ankit Halder. Ankit is integrated PhD student at IIT Bombay. He is working on using comprehensive multi-omics technologies to investigate one of the brain tumors, which is meningioma. And Ankit is the PMRF candidate. He is also utilizing this opportunity to interact with all of you and share his learning. He is quite strong in the computational and data analysis part. So do make use of these interactions with him, which is once in a week happening, every week, live session, so that you can also learn some of these software and data analysis tools. Let me introduce Ankit here.
Hello, my name is Ankit Halder. I am a third year MTech PhD dual degree student at IIT Bombay working in Proteomics lab under the guidance of Professor Sanjeeva Srivastava. My research topic is on one kind of brain tumor, meningioma, to identify the different altered proteome in the disease condition. So I am a PMRF fellow and is taking the live session on Introduction to Proteogenomics course every Saturday from 3 p.m. to 4 p.m.
All right, so you can interact with Ankit more often every week. The Proteomics course has 3 TAs, where we have Shakti Parihari. Shashwati is a PhD student in BSB IIT Bombay, where she's working on triple negative breast cancer as a part of her joint PhD program from IIT Bombay and Tata Memorial Center in Mumbai. And she's investigating in different multi-omics characterization to understand this complex problem of breast cancer. Let's hear from Shashwati about her work.
Good morning, everyone. Myself, Shakti Parihari. I am currently in 4th year of PhD in Biosciences and Bioengineering Department at IIT Bombay. I am currently working under the guidance of Professor Sanjeeva Srivastava. My PhD topic mainly revolves around multi-omics characterization of triple negative breast cancer towards identifying novel therapeutic targets in Indian population. So I will be your TA for MOOC course Introduction to Proteomics. I am happy to take any queries from your side. So let's keep learning. Welcome to the MOOC course. Thank you, everyone.
So Shashwati has also been TA at IIT Bombay of my Proteomics courses, which we teach live here, and she's quite experienced, good TA working in this particular area. So utilize her experience and expertise of Proteomics and also the teaching ability what she has. Next is Meghagayatri. Megha is also PhD student in BSB. She's working on medulloblastoma, one of the pediatric brain tumor, again along with Tata Memorial Hospital clinicians. And she also brings sort of her expertise working on proteomic technologies as well as cell culture-based experiments. So let me now introduce Megha directly from her.
Hello learners, welcome to the course. I'm Meghagayatri. I'm a third year PhD student here in IIT Bombay in Professor Sanjeeva's lab. My work revolves around a pediatric brain tumor called medulloblastoma. I aim at utilizing high resolution proteomic techniques to understand the disease better. I'm also working on developing a targeted proteomic assay for better diagnostics of the disease. And we finally aim at clinical translation of all the knowledge that we are getting from this.
All right, so you can learn many software tools, especially for mass spectrometry and targeted proteomics based workflows from Megha, and she's also quite experienced TA in proteomics course. Last, but of course not the least, is Sneha Rana. She's also a PMRF candidate working in the chemical engineering department under Professor Pramod Wangikar as a PhD student. She has been a student of my Proteomics courses at IIT Bombay earlier and did very well. So she's now going to work as a TA, which she's already taking some of the live sessions every week with all of you. So utilize again some of the learning experience what she has and clarify your queries. So let me introduce now Sneha.
Hello everyone, I am Sneha Rana. I'm a PhD student at Indian Institute of Technology, Bombay. I work under the guidance of Professor Pramod Vandikar in Chemical Engineering Department. So my PhD thesis topic involves unraveling novel biomarkers for diabetes mellitus using metabolomics approach, and we are using various mass spectrometry based techniques like LCMS and GCMS for our analysis. Apart from my PhD work, I like to read books and roam around in this beautiful campus. So thank you all for listening to me. And I hope to see you all for our next session. Thank you.
All right, well, so please interact with Sneha every week. She is conducting the live session. And again, she brings experience working in the same area herself about using some of the cutting edge tools of LCMS, GCMS to investigate metabolomics. So I hope by now you are already familiar with the MOOC, every week sessions and the videos and the way we conduct the course. Of course, these are recorded sessions, what you are watching. But in addition to that, we are also conducting these live sessions every week by the PMRF candidates, Sneha and Ankit, 3 to 4 p.m. on Saturday afternoon. Please use that time and opportunity to do the live, interaction with them and clarify your doubts. Also, we have once in a month live sessions directly from me and my team with the live from the lab, which is we are connecting today. And also we have one session in September when we will talk about another theme about integrated omics, how that can be utilized for the COVID-19 research. So while MOOC courses are online courses, but I hope you understand that, with all these interactive tools, we are trying to give you the glimpse of the way you would have taken this course directly at IIT Bombay live in my course or in my class. And it all depends on how much, interested you are to take your learning to the new level. And accordingly, we are there to help you out, right? So today's theme is essentially giving you the flavor of mass spectrometry based proteomics, but within the broader umbrella of the how omics technologies are really transforming the translational research, right? And that's where you will see that, in both proteomics and proteogenomics, there are many new tools, new technologies, but they're all aiming towards making some, impact for the translational research. There are many omics technologies which by now you are familiar that you are looking at gene level information in genomics, mRNA, transcript information in transcriptomics, protein based information, proteomics. Of course, you are also looking at some of the, if you are looking at biological samples, clinical sample, you look for those clinical features, and then you would add that information to look at the phenotyping, or that could also be clubbed into the metabolomics. And all of this information generates the big data sphere, which you can club as a big data. And each one of these, field which we are talking has so many instrumentation, so many technologies, which are quite capable of generating terabytes and terabytes of data every day. So generating data has become very easy, but of course, comprehending data, understanding data, it still remains very challenging. And that's why I think these courses are helping you now, and of course will help you eventually more to see like how to take these data sets and analyze the data set and make more meaningful insight from those clinical data set or even any biological data set for that matter. Now, since our theme is more on the clinical today, if this big data which we are generating from the patients or the clinical samples, that could be utilized for many applications, right? And one of the major application could be the personalized medicine. The way the medical field is now transforming, goal is not to generalize the treatment, but rather make it very specialized and personalized. And therefore, more precision medicine-based approaches are coming forward. The way we have been, everybody's blood test is done uniquely, right? Like if you have any fever or any problem medically, your, various body fluids are being measured to look at different type of biochemical parameters to assess your health. But when it comes to the complex diseases, it becomes very generalized, like diabetes or cancer or any other bigger metabolic disorders. Then doctors are applying the very generic medicines. Why not to do that more personalized? And that is possible if we utilize these technologies to generate the data from the individuals who are having those issues, and then use that information to look at how the diagnosis could be improved, in which way when the drug is being given, we can monitor the drug responses, is the patient responding well or not. Multi-marker assessment can be done. One could also look at targeted therapeutics approaches. So now you are looking at very specialized or the targeted approach of treating the patients rather than making it very generalized. And that's what I think these technologies are going to give us, at least one step further ability in that direction. There are many omics technology, but more specifically in proteomics, you have been taught so far about gel-based proteomics, which you have learned different type of gel-based technologies from SDS-based to the 2D gels to the dye gels, and we are moving to the mass spectrometry-based approaches. In the proteogenomics courses, you have been exposed more towards the technologies like microarrays and next-generation sequencing platforms. But of course, eventually we'll talk to you about mass spectrometry as well, so that then one could combine the two to make the impact of genomics and proteomics together to make it proteogenomic information. So there are many technologies and tools available. Of course, this session is not about the theory, which I don't want to spend too much time about it, but I'm just refreshing you that there are many techniques which we are going to cover in these courses, and some of these have already been exposed as of now. Today, we will just focus mainly about one of the technology which at the end of the courses you will appreciate yourself, that mass spectrometry based platforms have really made huge impact for various applications. Whatever sample you want to analyze, whatever protein you want to look at, you want to identify the protein, you want to quantify the protein, you want to look at the interactome with the pull down assays. Everywhere mass spectrometry based platforms are quite important. And not only proteomics, but even metabolomics, mass spectrometry based system can be utilized. So one approach which has really transformed the proteomics field is the shotgun proteomics. Aim was, while the gels are very good, 2D gel and dye gels, one could separate out a protein, but they're very cumbersome, very tedious. Why not we try to utilize, a shotgun proteomics approach where you don't have to separate the protein from the gel, but rather you take the complex lysate samples, digest those, make those peptides, separate those with the chromatography, ionize them with electrospray ionization, and those ionized peptides will move inside the mass analyzer, where you can separate them with a tandem mass spectrometer with different type of mass spectrometer configuration like quadruple time of flight, QTOF, or Orbitraps, or different type of ion trap. Many of these mass analyzers can be utilized to separate these complex ions. Finally, after running these samples, you generate these kind of mass spectrum, and that could be utilized to look at the the B and Y ions, and finally looking at the databases to identify the protein. One of the instruments which has been very crucial in this particular journey is Orbitrap technology, which has really improved the resolution of separating these protein. And this particular instrument, which we are going to show you shortly in our lab, is Orbitrap Fusion LC-MS, where when the peptides are getting ionized, these peptides are coming inside the mass analyzer, which we have here tribrid mass spectrometer system, starting from the quadrupoles to the Orbitrap and the ion trap. So you are utilizing three different type of mass analyzer property to make it very robust system, which can be utilized for the MSN, MSMS or MSN type of analysis for various applications. With our advancement happened in the mass spectrometry field, in 2014, first time the draft human proteome map was deciphered to reveal 70% of the human proteins. Next year, there was an antibody-based approach came forward to make the tissue-based human proteome map. And very recently in 2020, November, the high stringency blueprint of the human proteome was deciphered, which aimed to look at a very high stringent data analysis criteria, and it still revealed greater than 90.5% of the human proteome. It means we are getting close to make it 100% of gene-coded protein, for which now we have experimental evidence from the mass spectrometry. So this is the current status of this field. In the today's live session, we are going to take you to the different instruments and show you live from the TAs who are going to describe the instruments. The first one among these, as we mentioned, about Orbitrap fusion, which we are going to take you from our lab. This is one of the very crucial transformative technology which can be used for many applications. While the different type of simple and basic mass spectrometers, even including multi-TOF, can be utilized to identify the protein, like what protein you have from the given band or the spot or from the given sample. But when it comes to the quantification of the protein at the proteome-wide scale, it means we are talking about thousands of protein, and each protein we want to quantify their level at each peptide. So that's where these kind of instruments and these robust technologies becomes very important. So there are two approaches of looking at the quantification or the abundance-based proteomics. Now, let's say you have the healthy individual versus disease individual, which you want to compare. You can compare them in using different type of technologies, but in mass spectrometry approach, you can use them either with label-free or label-based approaches. Label-free approach is very simple. You are taking the protein samples, digesting them to make the peptides, and those peptides you are cleaning it up, running in the mass spectrometer to generate these kind of chromatograms, saving that data in the library, and then using that eventually for the data analysis. Whereas in the label-based approach, you have like healthy individuals and disease individuals, or control versus the disease conditions or treatment. You want to label them with some sort of tags. And in the course, we will talk to you about different type of tagging strategies like isotope coded affinity tag, ICAT, or iTrag, isobaric tagging for the relative and absolute quantification. The one technique which is still currently very relevant is TMT, tandem mass tags. So these tagging strategies are made such a way that in your control and treatment, you are not adding any extra mass. They are isobaric tags, and therefore you are going to have the same mass added into all of the condition. And then when you are doing the MSMS fragmentation, these reporter ions from the tag comes out, and that is being seen as the reporter ion which you can utilize for the quantification of the peptides. So these are some of the complex detail which is more theoretical, which you can talk and learn more from our course. But now let me take you directly in the lab where Abhilash is going to talk to you about Orbitrap fusion and how this instrument and technology can be used for proteomics application. So Abhilash, please show the instrument from the facility and talk about it. You're not audible yet.
So this is the instrument.
You're audible now.
This is the Orbitrap fusion we have. This instrument we can use for performing different mass spectrometry based proteomic analysis. Please know the instrument fusion. Yes. So here the Nano LC is coupled with the Orbitrap fusion instrument. That is why we call this as the LCMS, right? So once we inject peptide, we can do different analysis on the basis of the abundances of peptides. Suppose you want to perform some analysis between control and test sample, and you wanted to know what are the different proteins that are altered between the control and test sample. So what we can do is we can get the sample. So the sample preparation is whole different field. We can prepare the sample, and what we need is the peptide. So for that, in solution or in gel sample preparation we can do that. We'll discuss in the upcoming sessions. And once the sample is prepared, we have to just place it in the auto sample, right? Yes. So this is the auto sampler. So we can prepare a kind of this auto sampler in this plate format. We can take at least 96 samples at a single shot, right? So we'll place the sample, and then we can start the acquisition process. So in this Nano LC, the acquisition will happen, and these are the columns, the tubings of the Nano LC, where there will be a kind of this is actually the chromatography, right? So here we will do a chromatographic separation of peptides on the basis of the hydrophobicity of the peptides, and then through this column the peptides will get separated, and then they will be injected through the mass spec using this device. So this is the probe and or the source of the instrument where the ionization will happen, and subsequently the fragmentation will happen and then will detect. So on the basis of coverage and abundance of the peptide, we are using different softwares like Maxplant and Proteome Discoverer. We generate the protein of that particular peptide, and then we quantify and try to do statistical analysis, which will tell the differential expression of the peptides or proteins present in the sample.
All right, thank you, Abhilash. And so what you see in the front is a Nano LC. While you can use different type of chromatography, you can use a UxPLC. But since we are looking at peptides right now for proteomics-based analysis, so it is good to have the Nano LC so that you can utilize very less amount of your peptides, inject them, and you can still do the proteomic analysis. But of course, depending on your application, you can interface these instruments with different type of HPLC platforms. So next technology which we are talking is about Q-executive mass spectrometer, which is a hybrid mass spectrometer. What we just showed you, the most advanced mass spectrometer, one of the best in the world right now, Orbitrap Fusion. The next instrument we are going to talk about, Q-executive mass spectrometer, which is a hybrid instrument which contains two different type of mass analyzers from the quadrupole and Orbitrap. Of course, this instrument is also very high resolution, and one could do proteomics as well as metabolomics, both of these applications. But at least in our lab, we primarily use it for metabolomics to keep the instrument separate so that you don't have to keep changing the configurations. And this instrument, then we interface them with the UxPLC so that you can have the high flow rate injection for looking at your small molecules. So next we are going to have the TS from the lab going to talk to you about metabolomics application using LC-MS platform, especially Q-executive.
Okay. So this is Q-executive instrument. So Q-executive instrument is a hybrid instrument which has been connected with the Vanquish UxPLC. So let me first give you an idea how this UxPLC and the Q-executive actually work. So as you can see, these are the two binary systems. As this is the UxPLC, so obviously the solvents requirement will be pretty high. And this is solvent A, solvent B. So solvent A is mostly 0.1% formic acid, and solvent B is methanol. So this binary solvent system. First take the solvent from here, and as you can see over here, the pump is there. So this is the first mixture that will help the solvent A and solvent B to come and mix over here. From over here, it will go this particular tube. As you can see, this will go to the injector. And from over here, we keep the samples. So as you can see, these are each of the samples. These samples contains cuvettes, glass cuvettes, that is having the metabolite extraction samples. So on the basis of the position that we will give, the particular injection will come, and over here, this will take the samples. As you can see, the particular, the auto sampler can rotate accordingly. From here, these particular samples will go and will inject into this particular analytical column. And this analytical column is connected from this tube, and this will go directly into the source over here. And the spray will happen from that particular point. So once this particular spray happens, so this is how, a chromatogram actually looks like. And this particular chromatogram, as you can see, we generally, inject or spike one particular known metabolite, which is maybe razorpine. So we have to be very much sure that the particular spike in metabolite that we are using should not be present in the actual samples. So razorpine is an alkaloid that we get from the plant, and generally we work with human samples. We do that the particular samples will not be present in the human. So the first thing what we see is that, as you can see, the this particular retention time of the 9.55, and this is the 609.27761. So this is the a particular retention, the M by Z of the reserve time. And this particular retention time 9.55, we try to see whether it is coming in all the samples or not. So like this, we check all the samples one after another, and we try to monitor whether this 9.55 is coming or not. This is the initial monitoring that we do to understand the particular samples has been done properly, and the particular intensity that you can see over here is 3.476 intensities are coming. We also try to see the intensities in run to run to understand whether there is a run to run variation happens or not. So this is how a setup of a QExactive and the UxPLC helps us to generate metabolomics data for the global metabolomics data, and further if we have some metabolites from here which we identify, and then we can also use this particular experiment for making a targeted sim study. That means of that particular metabolites will only be taken forward and can be checked for more number of samples. So,
all right. Thank you, Deep. So you have just seen, like, these kind of instruments can be utilized for different applications, and targeted approaches can be then taken forward to validate your findings. So now we have talked about two different applications, proteomics and metabolomics, using LC-MS/MS based platform. Of course, this is only giving you the bird's eye view of using these instruments. But next, then, how to now validate these findings. You have identified some proteins which you are thinking as the potential candidates or candidate biomarkers. And likewise, you have done metabolomics and you have identified few specific metabolites which you want to validate. So now you can use different approaches of doing validation. While many antibody-based approaches are more powerful, but especially if you want to use mass spectrometry-based approach, triple quadrupole-based mass spectrometers are very powerful way of validating your findings at the targeted level. So what we are now looking at, targeted validation, a specific target which you want to validate, right? And that's where we are going to use different type of triple quadrupole or Orbitrap mass spectrometers. So two different approaches broadly have been used. One is known as MRM or multiple reaction monitoring. And here what you would like to do, you'd like to screen very specific peptides which you have identified from the screening work of the discovery work of any kind of deep proteome analysis or quantitative proteomic analysis, that you have the set of peptides which you want to validate. So now those peptides, you have used some software, especially software known as Skyline, when you define these peptide sequences and generate the specific transition list. And now you're only going to monitor those transitions of these peptides which is of your interest. You are going to remove thousands to millions of peptides which is still part of the sample, but you are not interested in every single thing, but you are especially looking at the peptides of your interest which has shown good differences between your control and treatment conditions, which has passed a lot of statistics and showed good fold changes. Only those you are taking forward. In a triple quadrupole mass spec like Q1 and Q2, in between there is a collision cell. You take this, let's say the blue peptide, and that is getting fragmented in the collision cell, and now you are in the Q2, you are generating MS/MS fragmentation of that, which is going to generate the translation of that particular peptide, and that is going to give you the intensity information over the time. So this is the way to look at developing your selected reaction monitoring assays. Alternatively, one could also utilize Orbitrap instruments to do the parallel reaction monitoring. It is slightly more advanced approach, where aim is to look at simultaneously all the peptides of interest, and then trying to validate those peptides using Orbitrap instruments. So here you have quadrupole and Orbitrap, in between there's a collision cell. A typical chromatogram and the transition looks like this for these kind of targeted experiment. The same concept can be utilized for the targeted metabolomics as well. While doing these experiments, there are many considerations which you need to follow. You need to be sure about that your the retention time is accurate. Your some of the peptides which you take as the standard peptides are known as synthetic peptides, which you chemically synthesize. You'd like to first generate your calibration curves from those to look at their reproducibility, to also determine sometime their LOD, limit of detection, or limit of quantification, LOQ. And then you would like to take the unknown samples and then try to look at their quantification across different sample type. So these are some of the interesting ways of doing the validation. Now, the third instrument which we are going to show you is the instrument which I'm talking about, the targeted proteomics based application is triple quadrupole mass spectrometer, TSQ-ALTIS, which now Meher is going to talk to you directly from the lab. All right, Meher. The same instruments, can be used for both metabolomics or proteomics. But here what you will notice that the HPLC is what we are using rather than the Nano LC, because we want to develop these tests in a very short gradient. So Meher, now over to you.
Hello everyone. So now, as just talk to you about the targeted proteomics, in which two basic techniques are majorly used. One is MRM and other is PRM. So he has already explained its basic principle. So just to brief about it, that targeted proteomics, we already had the target list of proteins or the biomolecules such as metabolites, and we aim to monitor only our target list of proteins. So here we have this instrument that we use for this kind of experiment targeted proteomics. We have a triple quadrupole Ultrix from thermoscientific, and which is connected with SPLC system here, which is again from thermo, and it has different compartments, which includes this pump compartment, sample compartment, and column compartment. And here we have this auto sampler where we keep our biomolecule like protein or metabolite sample, and in this auto sampler at a particular position we keep it, and then based on the software which are connected to it, we run the sample. So this is the software from again from thermo itself. So this is the Excalibur, as you might have probably for the discovery proteomics you might be shown similar kind of software. So once we have done all the sample lineup and alignment, we know its position and everything, then we line up the sample with some method. So this method is created based on the list of target peptides using the software Skyline. So from that we get different values which are required for preparing the method, and two of them are, for example, for MRF we need the mass spun charge value for the precursor ion as well as the product ion. So based on that, pair of transition, pair of product ion and precursor ion, that particular set is known as transition, and multiple transitions are monitored in this instrument. So once we have created the method and we have named the sample, and we set up the particular method and injection volume, and we run the sample. So over the time, when the sample is run, finally we get the data, and which we further analyze again in the same software from which we have taken the transition list or the target list. So this software, it is in like interface of the Skyline software. So here you can see different on this side, on the left hand side, there are different list of peptides. So this is our target panel and target list of the target list of peptides that we just like we had run in the instrument, and then we can see in different samples how the peaks are looking or how their trends are there. So as here we can see that this for this particular peptide, the peak is looking like this. So here the different these different lines are different color represent different transitions. And here we can see the trends of peptides in different samples. So here we can see that for this particular peptide, in the sample that is on left hand side, the abundance is less than the sample on the right hand side. So this way we can perform a comparative analysis of for the target proteins or peptides using this kind of setup. And then again we have some of the spectra which look like this, which we can say that these are not the good peaks. So and this way, based on our observation as well as the software, we pick up the peptides or the peaks which are best, and then we perform the further analysis using this software. This was an overview of MRM experiment using triple quadrupole marker.
All right, thank you, Meher. And again, the same instrument, but since we are using the high flow rate here, you can do these kind of tests in a very short time. While for looking at the discovery approach, one need to have the long gradient to screen everything. But now, since you know the peptides and the metabolites of your interest, you can do these kind of tests in a very short time, maybe sometime even 5 minutes to 10 minutes gradient is good enough to look at your peptides and the metabolites of interest. And of course, shorter the gradient, it will be more clinically applicable because then you can do more tests in that time. So let's move on now to the next set of technology, which will connect us with the genomics and some of the other, the broad high throughput technologies. So next generation sequencing and microarray based platforms, as well as some of the label free biosensors. So that is going to be the next set of technique which we are going to talk today. Of course, the students of the proteogenomics course have been quite exposed to the genomic technologies, but even in general, even the proteomics students also realize that without having the sequence and the blueprint of the life, it was not possible for us to make these databases which can be used for searching for the proteins, right? So that's where I think the two are interlinked very closely. And the next generation sequencing goes back, as theoretically from the Sanger sequencing. And then eventually there has been so much advancement over the decades so that one could accomplish the draft human genome project and the human genome map. And then eventually the next generation sequencing technologies of came forward, which has really made huge impact for the clinics now, when you can do a lot of genomic analysis, very accurate, very fast, and much more cheaper as compared to what one would have thought in the long back while doing the human genome sequencing. So from the lab, now Ayushi is going to take this session about ion torrent sequencing, and she's also going to show you this instrument and very briefly give you a demonstration of that. Ayushi, over to you now.
Good morning, everyone. So I'll be presenting about the ion torrent sequencing specifically. So here in the slide, as you can see, so this is the instrument is ion torrent. So this instrument detects the sequencing based on the pH. So if you remember how the sequencing happens, so this is the DNA strand, and whenever the new strand adds the new nucleotide, there's a release of proton ions. So this instrument sequencing principle based on the detection of that hydrogen ions. So here you see, this is the chip where you load all the sequencing reaction, and this chip has multiple wells, and each well acts as a pH detector. So whenever there is an addition of a new nucleotide, there is a release of proton ion, and that proton ion is getting detected here by the pH sensor, right? So here you see this.
Ayushi, we lost your voice.
Okay, so this well is representing one well inside a pH chip, and this the bead-like structure is having the amplification DNA. So whenever there is the addition of new nucleotide, this proton ion is getting detected here, and the signal is coming, and that's how the instrument knows, okay, sequencing is happening. Now the ion torrent instrument has multiple instruments. One is ion PGM, ion proton, and ion S5. So coming to the sequencing workflow. Yes. So sequencing workflow involves these steps. So first and foremost important step is the library preparation. So wherever first you have to extract the DNA, and from DNA you prepare the library. So whatever is your target DNA, that DNA you have to amplify it. And for that you add a lot of primers, and those primers will help in amplification of your target DNA. And this all of the reaction happens on 96 well plate. So after the amplification, you have to combine all the reaction into one pool, and then to create amplicons, you have to digest the amplicons. And for that, sticky and you will add barcodes and adapters so that you can multiplex a lot of samples. And that's how you create the libraries. Once the libraries are created, then the next step is the clonal amplification. So the clonal amplification is basically of emulsion PCR, where you perform the amplification of your DNA. So here you see, you create a mixture where you add your reaction mixture, like the library, and then all the enzyme responsible for clonal amplification and the emulsion oil. And here you see each bead will act as a template where your DNA will be amplified over the bead. So likewise, at the end, you will have a bead which has all the clones of your single DNA. And then once the clonal amplification is done, you can perform the sequencing. You have to load this the result of emulsion PCR into a chip, and then you can start the sequencing reaction. Now I'll show you the instrument here, which is used for sequencing. Okay, so this is iron current F5, which is used for the sequencing. Once you have the ISPs, which has all the DNA now, that those ISPs you have to load on this chip. So this chip I was talking about, this chip has a lot of pH measurements. So here you can load your samples, and the pH strip will act as a sensor which will detect the proton ions, and this chip will go here like this. So the this chip will go here, and then you can close the chip clamp. All of the reagents so this is the reagent cartridge where all of the reagents like the nucleotides are here, and you have to place the reagents here like this, and the wash solution, cleaning solution, everything is placed here. And then after closing the instrument, you can plan the sequencing in the software, and then you can also monitor the run throughout the sequence. That's the demo for the iron tolerant S5.
Thank you, Ayushi. It is, of course, takes a lot of time to do the experiment, but it was, a good glimpse of the entire process. Moving on to the next technology, which is protein microarrays, where you have, hundreds of thousands of proteins printed on the small miniaturized glass slide or cytosolic membrane to look for various applications, but what I have shown here, more clinical application about patient serum sample, and look for the potential autoantibody biomarkers. One could use these kind of high throughput technologies to look for the protein interactors, even various type of post-translation modifications, and many applications are possible. So let's take you again from the lab, and Arko is going to show you our microarray instrument setup, which can be used for doing these kind of experiments. While Arko shares his video, what we have here, like, with different type of arrays, which we have shown on this slide, looking at the human proteome arrays, which looks for all the human proteins, or even we are also looking at currently some of the SARS-CoV-2 viral proteome. So those kind of chips. But the fundamental concept remains very similar to the Western blots. The way you do the, in the membrane, you have the, for an antigen, you would like to look for the primary antibody, secondary antibody, and then detection. Likewise, in the protein arrays also, we have similar concept. You have the antigens printed on the chip. You float the patient sample when there is a antibody present, which is going to bind to the antigen on the chip. And then you are going to wash them off, add a secondary antibody like IgG or IgM, which is conjugated with Cy3 or Cy5. And those fluorophores could be scanned to look at these kind of images, which becomes very complex big data again to look for a lot of data analysis. So Arko is coming from electrical engineering background, and he's doing a lot of software development and device development, and looking at these kind of complex intensity analysis using his computational tools. All right, Arko, so you can show the instruments and talk about it.
Hello everyone, my name is Arko. I'm a PhD student of Professor Sanjeeva Srivastava. Right now I'm doing micro data analysis. Now I'm going to show you regarding the microarray scanner, how we usually scan the slides. So just now sir given the idea regarding the microarrays. So how the microarray chip look like. This is a microarray chip, you can see. So here actually thousands of proteins are printed on the chip, almost 40,000 proteins are printed on the chip in duplicates. So now this is a kind of a group plot arrays, the manufacturer from CDI laboratories. So this is a protein microarray. This is one work we are doing. So another chip I want to show, this is called peptide array. So all these slides are microscopic slides. So that actually you cannot see anything in your human eye. So we want a very good instrument to extract the intensity of the proteins, all this. So for that, the next thing is that we need to show something regarding the scanner. So this is the scanner. So this is GenPix 4000 scanner. Now I will show how we usually scan the slides. So this scanner is actually here actually PMT is there inside for the it's a detector. So it is practically the light sensor. So we cannot open it right now. Already one slide is there. So now I want to show this scan one slide. So you can see this thing now. I want to show this scanning, how this scan actually going on. So you can see this. Yeah, so now you can see this scanning already started. Yeah, so after this we scan this particular chips. Now I am to show the data, how we can extract the data. So this is where this scanning is going on. It is in red channel. There are two channels simultaneously. For 635, it is called the red channel, and 532 is the green channel. So usually for hoop plot arrays, we usually go for 532 for quality checking of the chip, and 635 for the autoantibody screening. Now I want to show you this how actually the chips after this scanning, how the chips usually looks like. So now you can see this is a complete image of a particular slide. So now maybe this kind of spots you can see, these are very intense spots. So can be taken as the biomarker. So finally we need to go for a long process of the data analysis in our programming language. Then we can think that which is particularly a biomarker like that. But how we extract the data? For that, one particular file which is called the GenPix array list file, that is called GAL file, which is important to incorporate on the spots to extract the data. Let me show you how the GAL file looks like. This is the kind of a circular feature indicator. So we usually call the spots at the feature, and these grids you can see, these are called the feature indicator. So we need to map it very carefully because we want to extract the data, accurate data. So for that, the grid placement is very important. It's manual work. It's like taking too much time to we given lot of time for that data acquisition. So now after that, we just need to go to the result section. So then we need to select this data. So if we select on the data, you can see the data is generated, and this data is called the GPR file, which is called GenPix result file. That is very important for the further analysis of the data. So now this which I have shown actually, that is called the hoop plot arrays. Now let me show you another slide that is we are working for. Yeah, so this is called the peptide array. So now you can see. Yeah, now you can see this is called the peptide arrays. Yeah. So here you can see this is the in the different format the proteins have been printed, and the same way we need to place the grids properly in this way. Now you can see the formation of the grids. It is practically this rectangular fashion the grid has been developed. Now in the same way, if we go to the result section and finally if we click on the data, the data is generated accordingly, and this data is actually important, and that is called the GenPix result file. And how the GPR file? It's an Excel file, Excel sheet. Now you can see this is the GPR file. All proteins are printed. You can see the block wise, there are different blocks, column, row. So according to that, you need to know that which proteins you want to take, which data you want. So everything is there in the particular GPR file. So this is all about the extraction of the data, and then we proceed into the all the data into the R programming language for the further data analysis, statistical analysis, and usually take the call which is the significant biomarker for this. So thank you.
All right, thank you, Arko. And so again, analysis becomes very time and the intellectual skill-based exercise, and that takes a lot of time. But let's move on to the next last technology which we're going to talk today. How to take some of these interactomic technologies to generate more kinetic information, not only that interaction is happening, but also to look at their on rate, off rate, and the strength of the binding and the kinetics value. So there are many platforms which are emerging, especially surface plasmon resonance has been one of the key platforms for label-free biosensor. But additionally, new technologies are emerging, and one very promising technology is biolayer interferometry-based approach, which is now going to be taken the live session from the lab about octet technology from Shalini. So Shalini, you can now just quickly talk last few minutes. You are not audible yet. Shalini, please unmute and then speak.
Hi all, my name is Shalini and I'll be talking about the BLI system here. So this is the BLI machine of Sartorial. And here if you see, these are the two sets of the plates. 96 well plates. One is for the sensor part, where we have already placed the sensors. So this is how the sensor looks like. And here, this is the tray which holds it. And below this, we have the rehydrating plate, which is again 96 well plate. So here, if you see, we have already added the solution so that the sensors can be hydrated, because the sensors that comes here to us are actually coated with a high sucrose condition, so that anything which is bound on the sensors, like on this particular one, we have protein K biosensor. So on this, we have sensors coated with protein K, and to avoid the degradation of these, we have a coating of sugar, concentrated sugar, so that it can be protected from any kind of damage for the middle of the protein. That's why we need to hydrate the sensor before starting the experiment. So here, this is the second place where we have added the buffers, samples, and this is the regeneration part where we add the regeneration buffer and hydration buffer, equilibration buffer. So here we place it in such a way so that these are properly set, and then we close it. And here, if you see, this is the panel which picks up the sensors and then moves into the sample identification or measurements. So now we'll go to the software part, where I have already said the things for better understanding. So here, if you see, this is the part where the sensor plate is being shown, and this is the part where the samples are kept. So here, this is the one which we have loaded, which we are going to load with the protein, with the antibody. So protein A is having the biochemistry to interact with the IgGs. So here we have put some IgGs, and here also we have put some IgGs. But here these two are blank, which are same buffer as we have kept here in the hydration buffer as well as in the buffer way. So let's now see how the experiment looks like.
Shalini, we can't hear you.
So it will. So the basic idea is that when the biosensors will first bind to the antibodies, and then we can see how they are binding. So basically, this shows all the association and dissociation. This particular instrument is good for identifying the kinetics, the binding affinity, epitope, and such kind of applications. So this is also used for identification of the whether the natural antibodies are present in the human body in the infected COVID patients, and then they try to see whether the spike proteins are having any kind of antibodies naturally present in the host patients or not. And then they also use DNI for identification of the partners for the spike protein, that is ACE receptor. It's taking a bit of time, so.
All right, Shalini, so I think maybe, we can, due to the shortage of time, we can close the session right now. Of course, we can take it any other live session when we have, more time opportunity.
Thank you.
Right. So, again, there are different principles of looking at the biomolecular interaction. Idea here is that you are using label free. You are not, you are trying to utilize those platforms to generate the kinetic information. And octet-based system, which is from the principle of biointerferometry, gives you some ability of looking at high throughput approaches like the way ELISA was being done in the solution form. And similarly, now you can do these kind of kinetics of these interaction in 96 well plate format with the solution based stuff. And the same solution could also be utilized for your other experiment. And these are some of the powerful points for these technologies, right? While there are always pros and cons of which technology can be more suitable for your applications. So finally, I just would like to close the session by concluding few points. The course is online, but still, as you can appreciate, that we are very much committed to make the course to give you the live experience, right? And you should be utilizing these opportunities to interact with us more often. Many of the students who have been toppers of the MOOC courses have come to our labs for getting further training. Some of them have done internship here. Some of them have also further, even joined my lab as a PhD student or even, MTech students. So these interactions have been very useful and, have been very productive. So if you like these technology, these courses, you do interact more so that we can, see how best we could give you further training and enhance your knowledge in this area. Next live session we will be doing again on 16 September to talk to you more about these technologies in a specific application context of COVID-19, a research which we have done recently. But last, I'd like to close here with this slide, where I'd like to emphasize that none of the technologies are perfect, none of the technologies are complete, because we are talking about the biological systems, which are really complex, right? And think about when biology happens, any life system works, there are many molecules which play very intricate roles of the dynamics of working from genes to RNA to proteins to metabolites to the phenotype analysis. All of these together, they drive the biology, they drive the clinical features, they drive our metabolic systems. So to understand just one layer of information may not be sufficient, and therefore integrating your knowledge from taking from genomics to proteomics like proteogenomics, or taking more functional information from proteome and metabolome from the same samples can be very powerful, and you should be looking into those approaches. And if you are more computational oriented student, you should also try to look at how you can use this information to generate computational or mathematical models to make more predictions for the unknowns and use the systems level investigation from these kind of omic technologies. So there's a lot of opportunities, lot of unknowns, and that adds a lot of flavor for you to venture into this area. I hope you have liked today's live session. If you have more queries, questions, do directly feel get in touch with me by writing the email or chatting on the forum, and we'll be happy to take your queries and talk to you further. Okay. Good luck, everyone. We will again talk to you in the next live session, and we look forward for more active participation from all of you. Okay. Let me thank all the TAs and our Proteomics Lab IIT Bombay team who have in a very short time given you the flavor of the most cutting edge technologies. Thank you everybody for joining today.
I'll close the session. Okay, sir?
Sure, we can close the session now. Yeah.
Thank you, sir.